As David talked about in his last segment, the Reef Restoration and Adaptation Program in Australia has got a number of components to it, and a number of really great people that are working as part of that team. And I've had the fortunate pleasure of getting to know much better over the course of the last year and a half Dr. Lena Bay, and she's become a friend of mine. And I'm so excited that you guys get the opportunity to hear from Lena. Lena's a scientist at the Australian Institute of Marine Biology and Marine Science. She's an evolutionary ecologist by training, and she is becoming kind of the go-to person within the RRAP program on that topic. So Lena will be talking to you now, sorting the wheat from the chaff.
Thank you very much. I can hear myself, so I presume you can hear me as well. Thanks, Tom. I did put on my introductory note that my lucky number is 23, which is also on my card, so come and find me if you're also 23. Now, coral reefs are truly amazing places. Let me see, am I clicking forward? Coral reefs are truly amazing places. We're all in this room because we care deeply about them. They punch well above their weight, only about 0.25% of the oceans, yet they are the home to about 25% of its biodiversity. And they support and nourish millions of people with food, coastal protection, social, spiritual and economic values. But since the 1980s, corals have declined worldwide. And despite its huge size and its gold star management, the Great Barrier Reef has not been immune. So over the last, particularly over the last 3 to 4 years, we've seen huge declines in coral cover. And so based on these recent experiences, the Australian Institute of Marine Science a couple of years ago got scientists, natural resource managers and government officials together to ask the question, what else can we do despite the conventional management that we are currently undertaking? Now it's clear that we need action on climate change. We need to continue doing what we're doing. But are there other things that we can start thinking about that might help the Great Barrier Reef maintain its value and continue to be the icon of coral reefs that it is today? Now, the discussions that followed were overwhelmingly positive. There was a real appetite and need for us to think about this further in Australia. We were a little bit surprised by this because, frankly, as Tom said before, it was not a question that we had talked about before. It was not something that we had even considered. And so out of these discussions, the Reef Restoration and Adaptation Program was formed. And one of the main objectives, or a main objective of that program, is to develop a prioritised, costed research and development program, not just for these novel interventions that we're talking about, but thought through in terms of both the production of those interventions and the implementation on the reef. Now, while we're not suggesting or we're not talking about actually doing it yet, what we're hoping to do in the program is, in the initial phase, to make a plan for what we would do, and in the R&D phase that follows, develop the knowledge to implement these new tools if and when we need them. So we have taken a no stone unturned approach. We have examined, or we are examining, a very large range of interventions. What you can see in this image is a picture, roughly a picture for each of the intervention methods that we're looking at. Now, these interventions both aim to prevent the stress occurring or help accelerate the recovery if stress and damage is occurring. And I've led the production of a report that outlines the background of all these interventions, and I'd like to say that if Tali describes the NAS review as a a rosé. I hope that mine will come across as maybe a slightly more mature red wine. It will come out in the next month or two, so I hope you'll take a moment to read it. What I'd like to talk to you about today are the interventions that are associated with seeding corals onto coral reefs. Now, there are two broad objectives when we talk about seeding corals onto reefs. You may either want to enhance the abundance of corals, and you could use local stock for this. You may also want to enhance the performance of the corals that you use for reseeding, either so that they're more able to cope with the conditions that are currently being experienced, or to make them better able to adapt to future predicted conditions. And at this point, I'm not the first and I won't be the last at this conference to pause a moment and pay my respects to the late Ruth Gates, who really led a lot of the thinking in this area. I'd also like to thank and acknowledge Madeleine Van Oppen, who really came about some of these ideas in collaboration with Ruth, and who's continuing to carry out their shared vision in Australia. So how might we come about these enhanced corals? Well, there are, again, two broad ways. You can either work with existing variation, already tolerant corals in the reef systems, or you can generate new variation through genetic engineering, for example. The existing winners we might identify through the bleaching survivors from any kind of stress. We have typically identified those in the lab, but with the recent frequency of bleaching events, it's become much more feasible to actually detect this from the reef itself. And one example here are some corals we've had in the sea simulator at Ames, where you can see that 3 individuals are highly bleached, but one is completely happy. This was after very severe heat stress. We can also look across environmental gradients with corals thriving and growing in hotter or more extreme environments, carrying variation for tolerance. So what I would like to do in the following is to give you a couple of snippets of the research that I've been involved in for the last couple of years that have made me a believer that, in fact, we can work with existing variation to enhance the temperature tolerance of corals through restoration and adaptation methods. Now, temperature tolerance is heritable. What this means is that parents pass their tolerance off to their offspring. So through interbreeding corals from a very warm reef in the northern Great Barrier Reef, where the temperatures are several degrees warmer, with corals from a central Great Barrier Reef, we showed that the resulting offspring were up to 10-fold more tolerant if they had one, or even better, 2 parents from the warm reef. And this is really good news, because it means that any intervention that we implement on a reef in terms of temperature tolerance is likely to be propagated to future generations. Now, I recognize that we need to move beyond the larval experiments that we've done to understand how temperature tolerance is passed on to juveniles, adults, and the next generation before we can be fully certain of this. But you don't have to travel hundreds of kilometers to find this variation. Even within habitats of a single reef, you can find this. So in a second study, we looked at coral populations on the reef flat and the reef slope at Heron Island, and the environmental conditions are quite different between these environments, being more extreme in the reef flat environment. And we found very strong genetic structure across 200 meters. We then reciprocally transplanted these corals, and we found very high mortality and reduced growth in both directions of transplantation. But not all corals died. About 10% of them survived, and about 5% did so with little mortality. And these corals, in fact, grew as fast as the native counterparts in that habitat. When we then examined and tested their temperature tolerance, we found that some acclimatization had occurred, and that the corals that had spent about a year and a half in the reef flat environment had enhanced their temperature tolerance. But this was only good to a point. At the more extreme temperature tolerance, the locally adapted flat corals were more tolerant and the slope ones couldn't keep up. And so what this tells me is that there is existing variation out there. We can harness this, but perhaps we need interbreeding so that we can really pass on those genetic effects to subsequent generations. So far, so good. It seems that the research that we've done, in fact, that has been done by many groups around the world, support the premise of assisted evolution. But if we start thinking about implementing this on the reef, what could go wrong? Well, there are many genetic considerations that we need to have the forefront of our minds, and one is associated with disrupting local adaptation patterns. Corals are adapted to all sorts of conditions, not just heat. And by introducing heat-adapted genetic variants into cooler but warming reefs, we may in fact find that they're more sensitive to those new conditions, perhaps cold. What happens if you have a cold snap on that reef? Will they all die? And if we select corals that are very well adapted to cope with heat, might they be less well able to cope with other conditions? We know that corals that associate with the different algal symbionts show this response. So corals that were reciprocally transplanted between warm and cool environments that hosted different symbiont types bleached at different times. Corals with the heat-adapted Durostinium symbiodinium bleached when temperatures were just below the historical averages, whereas we found the opposite response with those that were associated with the Cladocobium. They bleached when temperatures were above the historical temperature regimes. But what happens if you are looking at corals that host the very generalized Cladocobium symbiodinium within the temperature range that they are experiencing? What we did in this experiment was that we grew corals with Cladocobium with some background of Durostinium, which is in fact the most common symbiodinium association on the Great Barrier Reef. And what we found was that cold was in fact not so bad for these corals. They grew as well and they performed as well, if not better, under these sublethal cold conditions than the sublethal hot conditions. And so what this tells me is that being a little bit too cool is probably not too bad, and the benefits of enhancing the upper thermal thresholds will outweigh the disadvantages that you might experience during winter conditions. But what about genetic correlations? Are the individuals that cope well under one condition, are they the same that cope well under another condition? Do we have a jack of all trades is a master of none, or do we have a lot of specialization occurring? So we conducted another experiment to look at this. We exposed a large number of corals to 5 different conditions, 4 stressful conditions and an ambient control. And we were not so interested in the exact phenotypic expression in any of these conditions. What we were interested in was whether the individuals that came out on top in one condition were also the same that came out in another, or were they the ones that came out on the bottom? We were interested in the genetic correlations. And what we found was that these correlations, when they were present, were overwhelmingly positive. So corals that performed well under one condition performed well under others as well. All the traits that we examined were positively correlated with survival, and what this meant was that the corals that survived any condition, one condition, were likely to survive them all, and we measured a very similar response in all the traits that we looked at. So this is really good news because it means that the variation out there is positively reinforcing selection in the direction of increased heat tolerance. So the studies that I've shown you now, the results that's coming out from the Australian Institute of Marine Science, many universities in Australia and overseas, support the idea that we can identify temperature-tolerant corals and that we can utilize them to breed hardier corals that could be used for reef restoration and adaptation. But once you have these corals, how do you produce them? How do you propagate them? And how do you get them out there on the reef where they can make a difference? So in the Reef Restoration and Adaptation Program, we've taken an end-to-end approach where we are looking at not only the science and the mechanisms of how corals can become hardier, but also how we propagate them and how we deliver them to the reef. Now, there are a number of different ways. Everyone in this room have more experience than I in this, at propagating corals and delivering them to the reef. You can multiply your corals either through sexual means or asexual fragmentation. You can do this in the wild on the reef, or you can do it in land-based facilities. And we're just on the tail end of coral spawning on the Great Barrier Reef. And some of these clips from Twitter just show the diversity of approaches that have been employed over the last couple of months. We've bred a large number of species, some of them, a single one, new to science in the laboratory, but people have also been out on the reef collecting spawning slicks, rearing them in the wild and seeding reefs directly. My role in the Reef Restoration and Adaptation Program has been to examine to try and put together with my colleagues a research and development program around coral hatchery and aquaculture production. And in order to do that, I employed a strategy of first designing a mind map to get the overview of what was involved, and then a theory of change framework to work through the branches that arose. And for those of you that are not familiar with the theory of change framework, what you do there is that you start off defining the goal, and then you work backwards and identify what activities are required to achieve that goal, and then the synergies that exist among those activities. The advantage of this approach is also that you can timeline the activities, you can identify gates or stop points where you might want to proceed or terminate research that is delivering or not. And so what I've got here is not a brain scan of what I looked like about 3 days ago trying to transition from coral spawning to this conference. In fact, it was much worse. This is the result after a fair bit of OC aligning and colour coordinating. What this is a mind map of all the activities that we think you need to undertake to be able to deliver and produce millions of corals that could be used for restoration and adaptation activities. And for each of these branches, we've defined the goal, as I mentioned before. So what you can see in the blue areas are the activities associated with enhancing corals, either through the existing variation that I spoke about, through hybridizing species, or through genetic engineering. In the green area, we have the treatment. So these are the microbial manipulations or the stress hardening that can also help enhance tolerance. On the red area over here, we have activities associated with keeping corals happy and growing in the lab, both in the young phases or through out-of-season breeding. We include research programs both for the sexual reproduction and the microfragmentation. And in the bottom orange area are the activities associated with the field deployment. Once you've bred the corals, how do you get them out in the field? What are the substrates you might want to use that enhance the early survival? And on the top here, we've got various activities that are associated with the facility, the logistics of transport, automation, and also supporting activities like cryopreservation, omics support, and so on. Now, from doing this, it became quite clear, and from going through and timelining and costing this, it became quite clear that we couldn't do everything, and certainly not at the same time. And so the second thing that I did was to go through and try and prioritize what we might propose to do first. Now, this is not saying that we don't think that everything in this diagram is not important and should not be done, but it's an attempt to say what should be done first and what do we need to put most emphasis on in the first couple of years. And the way that we did this was that we first identified the real showstoppers. We need enhanced corals. We need to be able to multiply these corals that are better able to cope with the conditions or the future conditions on the reef. We need to be able to keep them either in the lab or through these field-based facilities and make sure that they're healthy and happy. The second thing we did was to then look at the big cost drivers. So what are the areas where we could really drive down cost and enhance efficiency? And those areas were associated with field deployments. Is there anything that we can do to lift up the type 3 survivorship curve of corals to make sure that we have greater success per unit deployed? And then lastly, a big cost driver is your infrastructure utilization. So in order to ensure that we could use our facilities all year round, we are emphasizing both sexual and asexual reproduction. So it's clear that we still have a lot of knowledge gaps. There are a lot of things that we don't know, not only about heat tolerance and local adaptation, but also about the risks associated with these approaches. And it's clear that many of these are common among species and ocean basins. So really, to make great progress in this, we need international collaboration. It's clear that we need to innovate and we need to apply technologies from other areas. We need to get much better at what we're doing and fast. We need to be more cross-disciplinary than we have ever been. And lastly, we need to engage and communicate not just with ourselves in this room, but with a broad range of stakeholders that include tourism and fishing industries, Indigenous people and the general public. There's no doubt that coral reefs are facing a tough future, but we have an obligation to give it our best shot. Together, we can create new hope for coral reefs. Thank you.